Cancer. It's one of the most devastating diseases because it's likely that half of us in this room will personally deal with it in our lifetime. And sadly, for many types of cancers, there are no effective treatment options. Over 600,000 people in this country will die from their disease in this year alone. This is unacceptable, which is why my research aims to find better ways to help treat the untreatable. You see, cancer is the uncontrolled growth of cells. And we know that there are just a few proteins that cause these cells to grow and grow and grow. One of these proteins is called MYC. And I studied this protein because 70% of all cancer cells are full of it, and we know that it's critical for tumors to grow. So when I delete MYC in cancer cells, the tumors disappear, making it a perfect target. However, over the course of 30 years, scientists have not been able to find a way to target MYC. So the way that MYC works is that it binds to genes and either turns them on or off. I have found that MYC controls thousands of genes in cancer, but it can only bind to some of the genes that it actually controls. So we hypothesized that there must be another way that MYC is controlling these thousands of genes. So when I deleted MYC in cancer cells, I found that these tags, these red marks on the DNA, dramatically changed, meaning that they must be important because the cancer cells stopped growing. When I deleted MYC, I found that the tagging proteins that are responsible for setting these marks on the DNA were no longer being turned on. It turns out that MYC binds directly to these tagging proteins and turns them on so that they can tag the entire genome, causing genes to be turned on or off in cancer. So even though we cannot target MYC directly, in my research, I have found a way to target MYC indirectly by drugging these tagging proteins that MYC requires for its function. Thank you.